Hello everyone, I am here for this video task where I will be giving uh, answers on the based on biology as it is not of my background, but somehow I will try to give answers of my on my own, then check whether I have given the correct answer or not. After that, we will check all the incorrect answers through Google, then whether it will enhance my learning capacity or not. Then after again, based on the learning that I will achieve using Google, I will give the test again and check how many responses I will be able to give correctly. This video will be a quite long video. It will be around 1.5 hours to 2 hours. So there will be two breaks. One will be after giving the first test, which will be of 5 minutes. After that, I will there will be self-learning session using Google, then I will have to, then there will be again another break for 10 minutes and followed by the another session of my trying to whether I have understood the topic well or not. So without any wasting any time, let's start. So here it is our image, which is generally of plant cell. Let's check whether I am able to give answers correctly or not. So it is first question is that it is asking the component of a plant cell which is labeled as 6 represents an organelle that produces sugar molecule which is called, and here the 6 is given to this, and I somehow know that the sugar molecules are produced by chloroplast, so without wasting any time I will be taking on that. The level 4 represents and which is necessary for level 4. It is a very vast body covering most part of the cell, so it will be somewhat central vacuole, and which, according to my knowledge, it is somewhat helps in holding nutrients. So I am going to click on this. And level 10 is the internal layer the of a substance that surrounds the cell. This is called the cell. 10, it is this. It is somewhat looks like a cell membrane to me, so I will go with cell membrane as here it is written the initial as cell. So I am going with membrane. Okay, the function of smooth endoplasmic reticulum represented by label is. So endoplasmic reticulum, which will be somewhat, let's see the options. It is 2 or 9. Let's guess what will be the correct answer. It is 2 and 9 is over here. It might be 2 according to me. I don't remember it as of now. It is 2 and it is somewhat helpful in protein synthesis. So I will go with option 1. Here label 5 is present in. What is label 5 over here? Label 5, as you can see, it is a cell membrane itself. So cell membrane is generally present in plant cell, I guess. So I will go with plant cell only. The organelle labeled as dash will be in higher numbers in muscle cells compared to fat cells. However, this one number, as we can see here, it is present in many parts, and one, and what are the other options? 5, 8, and 10. And which is 5? 5 is this. Okay, 5, and other one is 8 and 10. 8 is a single component, so no, and 10 is also there. So I am going with 1 over here based on our guess. So we'll check whether it is correct or not later in this video. So coming to the next part. So the backbone of a polymer of nucleotide contains ribose sugar, What is the polymer? Actually, ribose sugar is generally present in RNA because as in DNA, it is deoxyribose sugar. So I'm going with RNA. And here, DNA monomer is called a nucleotide and is made up of three parts: a nitrogenous base, a ribose sugar, and a sulfate group. Which part is correct? Actually, DNA consists of nitrogenous base along with deoxyribose sugar and a phosphate group. So here in this case, our ribose sugar and sulfate group is wrong, so it will be nitrogenous base. Now coming forward, we can see here why is RNA not the main carrier of genetic materials. So it may be just what I can say is RNA is a stranded, so it carries half the information. RNA is less stable than DNA. USL is toxic. No, USL is not at all toxic. RNA cannot form long chain. So I guess it will be RNA is a stranded, so it carries half the information. Your justification, your justify your answer, and as it is, I'm going to use justification as this only, as say RNA is single stranded, so it carries half the information. Now moving forward, so here we can see there is a sequence of A, delta, epsilon, and T, and it is a single stranded, as we can see in the image. So according to the strandedness of this molecule, what type of nucleic acid is? So I guess it is a single stranded, so the option will be RNA. Let's check whether it is going to be correct or not. According to the types of bases in the molecule, what type of nucleic acid is? Okay, so here we can see A is present and T is present. But in RNA, T is not present. It is uracil in case of thymine. So I guess it is of DNA, but here it is according to, so I'm going to change this option to DNA, and here is as well, I'm going to give the answer as DNA. So what is the paradoxical about this image? So this image contains of bases like A and T, of not U. So this is a DNA molecule. So I will give the answer as in the image. Thymine base is present. Therefore, the given strand is of DNA. I will check whether it's correct or not. So according to your answer to question 11, what must be the final part of the molecule? Phosphate, nitrogenous base, and as I have clicked over here is DNA, so it will be deoxyribose sugar according to me. And according to your answer to question 12, what must be the final part of the molecule? Phosphate, nitrogenous base. So here I have also again selected as DNA, so it will be again, according to me, it will be deoxyribose sugar. We will check whether it is going to be correct or not in the future. So use the following information along with the previous to answer the following two questions. Let's see. Like what are the delta and epsilon more likely to be? So as we can see here, this A and T, which will be bounded by G and C. G and C will be on other side. So according to me, it will be somewhat A and T respectively, as it is of DNA molecule. We can see the nitrogenous base is of DNA molecules. So now we are moving ahead. What conclusion can be drawn from your findings? A genome that is comprised of single-stranded DNA is as sufficient as low double-stranded. I don't know whether DNA exists in single-stranded or not. So according to me, this is not going to be the correct option. An organism's genome needs C and G to reproduce. The type of sugar doesn't matter. Actually, it matters in characterizing whether it is DNA or RNA. This must be an exception to Kargas rule. Actually, I don't know about Kargas rule as well, so I'm going with option B here, which is an organism's genome needs C and G to reproduce. So coming to the next part, reverse transcription is the inverse process. Okay, as per I remember, transcription is somewhat RNA to DNA to the ordinary simple term of biology. It can be represented as so as transcription is RNA to DNA, so reverse will be somewhat DNA to RNA. Let's check like this. We will check further whether it is correct or not. The inverse of the process described in the previous question is, as we can see, the inverse of this reverse transcription will be transcription. So, option will be transcription. Which statements about protein is correct? A straight chain of amino acid is indicative of primary structure. Yeah, it is correct. A beta spiral is indicative of secondary structure. No, I don't think it is of secondary structure. A protein binding with another is indicative of tertiary structure. No, I don't think it is also correct. It is not correct. Quaternary structure is completely impossible. No, it is somewhat possible based on the. It might be correct. The but according to me, the most logical answer is the straight chain of amino acid is indicative of primary structure. Okay, so here as well, I will give the answer. So let's move to the next part. So using this diagram, we have to answer the following three questions. So let's check whether what are the questions. So our DNA template strand contains this sequence 3GTA, TTG, AAA, GCT, GTG, and 5 dash. What is the complementary strand of mRNA? So complementary strand will be starting from 5 dash to 3 dash. So whichever option has 5 dash to 3 dash. The opposite of G will be C. Okay, these two have the C option. T will be A. Okay, G will be C again. I have wrong. Okay, let's check whether. Okay, then for RNA it is string. So where we have achieved T will be A, C will be G. Yes, C will be G. But here it is not given like that, and G will be C. A will be U, and I guess there is a slight mistake over here. This one will be thymine will be connected to A and C will be connected to G. So it is somewhat here in both the cases we can see it is A. Just okay, moreover, let's see. But this confirms that G is connected with C. U is correct. Only this is what the difference is causing. So I guess C option is more likely to be correct. What is the more likely reason that the sequence in the question has been given written in reverse order from the answer? It is customary to do so. No, it is not customary to do so, but it is easier to understand in this way. No, it is not correct as well. The template strand is read 3 to 5, 3 dash to 5 dash, and the mRNA is synthesized 5 dash to 3 dash. Okay, this should be the correct answer. So, what protein sequence does this mRNA code for? So, here we can see from this image, and it is asking about this mRNA. So this is the mRNA. So we will see where it is CAU, AAC, UUU, CGA, and CAC. Let's check whether I am guessing it correctly or not. We will check whether we will get to know in the future about this when we submit the answers. So we can see that CAU, where it is histidine. So I will write histidine hyphen AAC. Where is AAC? AAC is over here. It is ASN. Let's write ASN over here, followed by UUU. UUU is where it is PHE. I will write PHE. It is CGA. CGA. Where is CGA? CGA, CGA, CGA. Yeah, it is here. It isn't design ARG. So I will write ARG hyphen and rest CAC. CAC where it is CAC. CAC we can find over here. It is history. HIS. Okay, so question in this segment is also completed. Let's see the next question. A point mutation has occurred, but the polypeptide is the same. Oh, I don't know about this. Silent nonsense, missense, symbol. So I am going with uh, silent. I don't know about this. I'm not from the biology background, so I will check whether it is going to be correct or not. Further, I'm going to answer this. Point mutation occurs in a separate polypeptide. The second one interacts with the first one in an epistatic manner and causes the original peptide to become truncated. I don't have any idea for that this as well. So I'm going with silent again. So let's submit and let's check whether it is correct or not. Okay, let's see what are the scores. Oh, it is somewhat 14 out of 25. It is somewhat good for a student of non-biology like background. So we here we will take a short 5 minutes break. Then we will learn what, as we can see the correct answer is provided over here as well. We will check through Google as well. So I will be going for a 5 minutes break. We'll come back and then start it once again. So here I am setting a timer for 5 minutes. So we will meet after 5 minutes. So, welcome back. The timer is over. So, now we are going to check the answers provided by me were correct or not. And if it is incorrect, then we will search it on Google and try to get the correct answer and then further apply it in the post session where we will be answering, where we will be testing our knowledge based on this searches. So let's continue. So here is the plant cell. The first question was correctly provided by me, which is label 6 represents an organelle that produces sugar molecules. What is it called? So it was chloroplast. However, I have only answered the second question. So let's check whether I will get the correct answer using Google or not. So first of all, I will be going to upload this image file over here. Okay, so it is. I have uploaded it. Now I will check for the question. Okay, so it is giving many of the photographs of this plant cell. Let's check the most relevant, which will be helpful for us. I guess this is the most relevant according to me, as it is giving a lot of detailed details in the image. Okay, it is somewhat a kind of book which will be around 88 pages long. So let's check whether we will be able to find that study mates. Okay, it will be taking a lot of time. Let's try. Okay, here it is. So it is showing it animal cell, but I guess it is a plant cell. It is a plant cell. Let's see what the level 4 is. Oh, okay, level 4 is this. So mm, we can't see distinguish. Okay, so here we can see animal and plant cell. Here it is a vacuole. Okay, this is a vacuole which degrades and recycles macromolecules, stores metabolite. Okay, so let's see whether it is in the option or no, or not. So it is showing holding nutrients, maintaining turbidity. Let's check whether what the turbidity means, as I don't know about this. Turbidity is a state of being turbid or swollen, especially due to high fluid content. See, that's it or being swollen, especially in plant cell. It is a condition that occurs when plant cells are filled with water, fluid content. Okay, let's check whether it is going to fulfill. No, it is not that much clear. Okay, we will be searching for some other photograph. Okay, we can see that, but it is all shade from, so we can't adjust that. We have to go for some board with some kind of open source. Okay, let's try this one. As we get to know that this is a vacuole, from that previous search. Okay, this is somewhat a question answer type, so this will not be helpful for us. So let's close this and I will be looking for this. Okay, hope so that it will provide me some better understanding based on this. Okay, it is describing every part as well. Let's see, is there any heading with vacuole? Yeah, here it is. It is like in tree structure in the cytoplasm. It's called vacuole. It is could be single and big. Okay, as we can see, it is large. Storage sacs for solid or liquid content. Central work, potato, no. Okay, here it is written. In plant cell vacuoles are full of cell sacs and provide turbidity, swollen and distended or congested, and rigidity to cell. So here we can see that it has provided a correct answer for this. So we will obviously going to apply it in the next session as well. So now let's moving forward. Levitin is the internal layer of a substance that surrounds the cell. This is called the cell membrane. Okay, I'm written here. So I have to look for the main function of the smooth endoplasmic reticulum by label and what it should and what its role is. So I'm going again to that this one. Okay, here it is written endoplasmic reticulum. We are going to look into this only. Okay, here it is categorized as well. So the question is describing is about smooth endoplasmic reticulum. So we can see here it is smooth. The SER helps in the manufacture of fat molecules or lipids important for cell function. So here we can see, okay, lipid synthesis, not a protein synthesis, it is a lipid synthesis, but what will be the labeling 2 or 9? We will get to know it from the image, which is here. Okay, so smooth endoplasmic reticulum is over here, and it is represented as a small bean-like structure. So let's come to the image in our question. Okay, it is a small bean-like structure, but it is not that much. Okay, here it is a small bean-like structure, which is 9. So the Google is able to answer this correctly that it is of 9 and lipid synthesis. So moving forward, the label 5 is present in. What is label 5 over here? Let's check where it is. Okay, it is label 5. Firstly, we will check with that image what this is. Cell wall, adjacent cell wall. Okay, what the question is again. Level 5 is presenting. Okay, we have to look for a cell wall over here. Okay, cell wall is absent in animals, plant cell in addition to the plasma membrane. Okay, it is not able to give the correct answer for us. Okay, we will copy and paste the whole question here. Mm, Label 5 will be cell wall. Is it cell wall exactly? Let's check it again. Okay, it might be plasma membrane. 5. It is not 5, it is 10. It is cell wall. Okay, we will check this open source. Okay, wait a minute. I am going to paste it over here. I have seen the exact match of that picture. Let's see whether I am able to find it again or not. Okay, here it is. Okay, here, from here, we will see what 5 number is. No, it is not written over here. So it is making a little bit challenging. Should I try it for adjacent cell wall? Let's check over here. No, it is not able to give the clear answer to me. So let us move forward, checking more sources. Okay, here we can see that which of the following is found in all prokaryotic cells, but only some eukaryotic cells. So it is somewhat cell wall. So, okay, so in this way we can see some prokaryotics and some, but however, it is a wrong option here as well. Like it is saying that all prokaryotic cells have a cell wall, but only some eukaryotic cells have cell wall. So there is somewhat, we can say, discrepancy in the question. So further, the ordinary level will be in higher numbers in muscle cells compared to fat cells. I have chosen one. What's one? Let's see what one exactly is. Actually, I don't know what one it was at all the guess that I have given. So it is mitochondrion. Let's see, is there any description about that which is helpful in giving the answer? Mitochondria. Okay, it is mitochondria. Here it is not written anything. Okay, we will search it on my own, on our own in Google. As we can see, it is mitochondria. Where is the question? Okay, yeah, you can just what the exact example spelling is. We will get it from here. Okay, Mitocho. And okay, let's check whether it is correct or not. Muscle cells contains more mitochondria because they have to release large amount of energy. Okay, from here we can see uh, mitochondria will be in higher numbers in muscle cells. Yeah, it is the correct answer that is given by Google as well and by me as well. So here the come to the next section. The backbone of a polymer of nucleotide contains ribose sugar, which is this polymer, which is correct, as it contains ribose sugars. A DNA monomer is called a nucleotide and is made up of three parts: a nitrogenous base, a ribose sugar, and a sulfate group. Okay, no, it is not the case. It contains nitrogenous base, a deoxyribose sugar, and a phosphate group. So the correct option is nitrogenous base. Okay, here, why is RNA not the main carrier of genetic material? Let's see the correct answer for this. We will use this tab only. Okay, why are those genetic material? Let's see the answer. Okay, it is a paid soft paid site, so they will not be able to see the answer here. Let's go for some better, like it is an open source. Like its RNA is very reactive molecule. Our genetic material should not be very reactive. They should be remain stable. RNA undergoes hydrolysis almost times faster than DNA. DNA gets repaired faster than the RNA due to the presence of cytosine. No, it is not. Okay, this will be the more suitable answer, I guess. Mm, no, is there any site? Yeah, here it is. Okay, we can see here DNA versus RNA that in case of RNA, two OH group present in them make them more reactive. Hence, DNA is proved to be less reactive chemically and more stable structurally than RNA. Okay, so the answer is RNA is less stable than DNA, which is correct over here. We can see that DNA is more stable structurally than RNA. Moving forward, we can see justify the answer. Okay, here we can see that it is written that RNA has two OH group present in them make them more reactive. It is a similar explanation which is used by this. RNA has one more hydroxyl group than DNA. As such, it is less stable. So this is also easily provided by Google as well. So coming forward, let's upload this image and see whether it is able to provide the answer for this or not. So I've already taken the screenshot. I will have to copy the answer. So sorry, question, and then search it over here. Okay, we can't see the exact match of this image. So let's see using this image only whether it is going to allow me to read or not. Oh, it is a book, so it doesn't allow me to read through it. Let's check this one. Okay, this is not the question that I was expecting over here. Okay, so let's move forward with some other image. I don't think it is given over here. Any question like this, all are totally different. Okay, leave this question. We will move ahead. Okay, according to the type of basis. Okay, let's see the question as well. According to the strandedness of this molecule, what type of nucleic acid is it? Okay, it is a single stranded showing, so it might be due to this reason it is RNA. Okay, but I'm not able to get the answer from Google, so leave it here. So the other one, according to the type of basis in the molecule, what type of nucleic acid is it? So as I have said that there is a presence of A and T. Therefore, it is a DNA molecule. If in place of T it was used, it will be RNA. So this is correct in my case. What is paradoxical about this image? Okay, as I have said that it is a DNA, but the correct answer is nature indicates RNA, while type of basis indicates DNA. However, first part was not be able to provide was not be able to be provided by Google, so we will leave it here only. So according to question 11, what will be the final part of the molecule? So as it is RNA, so it will be ribose sugar, but I have ticked it as deoxyribose sugar. Then, according to your answer to question 12, what must be the part of the molecule? So I have actually guessed it as DNA. So deoxyribose sugar is the correct answer for this as well. So if this is correct, so we don't have to learn ourselves. So it is correct. What are delta and epsilon most likely to be? As it is DNA, so it will be A and T respectively. It may be GMC also, but no option is given based on that. So it is correct, A and T respectively. What conclusion can be drawn from your findings? So I have taken it as an organism genome. I will copy and paste it and check whether it is able to give me the correct answer or not. Okay, I'm not be able to find this. Actually, it is showing Karga's rule only. It has to be based on my findings, and as we don't be able to submit a PDF in this, so we will have to move forward. Okay, I have given it wrong. Okay, let's check it on Google as well. So reverse flow of information, central dogma. Let's see, is the answer given? No, it is not showing up transcription, reverse transcription. So we will get here. Reverse transcription is a process of using mRNA to produce two complementary single-stranded DNA. Okay, single-stranded DNA is also present. So in this case, the previous answer will be the genome that is comprised of single is sufficient. Let's check whether it is true or not. We will check it out here. Okay, we are not able to get the correct answer of this. Okay, let's carry forward. So reverse transcription. So let's see. It is using mRNA to produce complementary single-stranded DNA. So we are doing it RNA to DNA. But I have marked over here. Okay, it is wrong by me, wrongly answered by me. As we can see here as well. Okay, we can see here as well. It is reverse transcription is RNA to DNA. What I have given, it is DNA to RNA. So it is wrong in my case and correctly given by Google. The inverse process described in the previous question is actually it will be transcription. It's totally based on the common sense. Which statement about protein is correct? Let's check it with Google. Okay, where we will see. Let's see with Baidu. Okay, here we can see that protein structure are made by condensation of amino acids forming peptide bonds. The sequence of amino acid in protein is called primary structure. The secondary structure is determined by dihedral angles of the peptide bonds. So here we can see, no, it is the dihedral angle, which indicates the secondary structure. And this one is correct. Amino acid is indicative of primary structure. This alpha helix is that secondary structure. What is tertiary structure? Is the folding of protein chains in space. What is here? Protein bonding. No, it is not. It is the folding of proteins. Let's see what is quaternary structure here. What is quaternary structure? Two complex functional proteins result in. Okay, this is of no use actually, but it has helped me to get the correct answer, and my correct answer was already correct. So let's move ahead for this. Actually, I have found one discrepancy in this question, like G's will be replaced with C, T with A, G with C, T with A, C with G, and G with C. Okay, is all right up to here. A will be U. Okay, sorry, I'm going backward. Okay, this G will be C, T will be A. No, I'm really, really sorry. It is 3 dash will be 5 dash. This G will be C, T will be A will be U. Okay. T will be A, T will be A, G will be C. Okay. A will be U, A will be U, A will be U. It is also okay. This G will be C, C will be G, T will be A, and G will be C, T will be A, and G will be C. It is actually full correct. Okay, I'm sorry for that discussing about the discrepancy. It is correct. Okay, what is the most likely reason that the sequence in the question has been written in the reverse order from the answer? However, I have get the correct answer, but let's check with the Google as well. So the sequence of is transcribed. There are two types of. No, it is not. It is not giving the correct answer. Okay, let's go to images. See if it is helpful. Okay, here it is. Okay, it is giving something. RNA polymerase, the enzyme responsible for synthesis RNA, can only add nucleotides to the third 3' end of the going strand. Synthesis, RNA synthesis must proceed in a 5' direction. Okay, simple. The template strand is the 3' to 5'. Okay, it is correctly given. Okay, so from here we can see that Google is that Cura on Google is able to give me the correct answer as it was already correctly given by me. And what protein sequence does this mRNA code for separate each protein with hyphen? So as we can see here with this, I have matched the table with this and get the correct answer. So I don't have to search this on Google as I got the correct answer for this. However, this question was totally new for me and I will check it with the Google whether it is giving me correct answer or not. Okay, a point mutation has occurred, but the polypeptide is same. What is the mutation called? Okay, polypeptide means the same amino acids. Let's see. Okay, it is giving that silent mutation code as a correct answer. So, however, I have guessed it correctly, but I didn't know anything about this. But Google has helped me to get the correct answer by a proper description, like single silent mutations code for the same amino acid, and it does not affect the functioning of the protein. So moving ahead uh, with the last question, I guess. Let's check whether I will be able to get the answer from Google or not. So here it is. Is there any chance by we can get the answer from Google by use? No, it is not. Let's check with other part as well. Okay, here it is not given anything. Let's check it over here. Hmm, no, it is not. It will not be very helpful for us to get the correct answer. Let's paste the options as well. Okay, here we get the Baiju's. Okay, from here we can see the key differences. It will be very easy to get the difference between missense and nonsense mutation. Okay, as in question it is written like a point mutation occurs in a separate polypeptide. The second one interacts with the first one in an epistatic manner and causes the original polypeptide to be truncated. Okay, so let's check. Can we actually, okay, stop codon notation site. Okay, it's different. No, no different amino acid is substituted, so it is in epistatic manner, so it is getting it correctly, and it is a truncated protein. So we can see here as well, it is becomes truncated, so it is a truncated protein. So Google helped us to get a clear difference between nonsense and missense mutation. So now I will be having a short break of 5 minutes. Like we got all the answers over here in Google, but somehow we will not be able to get the answer for this part. We will be answering it in the same manner as it was before. So we will check our knowledge whether it is enhanced or not in the later session. So I will be setting a timer of 5 minutes for a break. After that, we will be again meeting here. Okay, see you after 5 minutes. So welcome back. The timer is over. Okay, so now I am going to have the second part, which is let's check whether okay, it is totally different. Let's see first of all what is going over here. After a short break. So now we are going to test our knowledge based on the retest and after learning through Google. So now I am going to test my knowledge by using this set biology A. So let's start. So here it's taking a little bit time. So let's wait a moment. Okay, so we can see here the image is totally different to that of retest and the questions that we have searched on Google to increase our understanding. So here we can see that it is not a plant cell structure, it is a animal cell structure. So let's let it load first. Okay, so here we can see the first question is label G represents several proteins that assist with cell division, production, and maintenance of cell shape. What do these proteins comprise? So as we can see, G, this is what we are looking for. And from the plant cell structure, if we see, there is also this kind of a structure. So the right option for this is cytoskeleton. Now further, what this label F represents, which is necessary for, and the F is here this, which is nothing else but centrioles, and it is necessary for cell division. So here, this will be the correct option as per my knowledge that I have using gained during the self-study. So moving ahead, the ordinally represented by label D is mainly found in, and the and it is saying specifying that our answer should be the names of the organelles. Separate your answer by a comma and then a space. So what is this? It is label D. So label D is somewhat here. We can see that it is nothing else but uh, we can say a ribosome, as this dot-like structure is ribosome, and this portion is nothing else but, as we have seen in plant cell diagram, it was what we can say smooth. No, I am not getting it right, so it might be found in like smooth endoplasmic reticulum was not this type of. It is none other than like yeah, it will be smooth endoplasmic reticulum, and we have to separate it within comma and then space. Okay, so this is my answer. The main function of the Golgi apparatus represented by label is, so here we can see that what the Golgi apparatus looks. So this packet-like of structure is somewhat called Golgi apparatus. Let's see, is there any option K? Yeah, it is K. And it is helpful in lipid synthesis. So the option is K and lipid synthesis. The organelle label C is present in what is C here? It is a, we can say, as we are well aware of, it is nucleus and this will be nucleolus. So is there, and it is present in both plant cell and animal cell as well. So the correct answer is animal cell and plant cell. And here it is the organelle labeled will be in higher numbers in muscle cells compared to fat cells, as we can memorize from the part A, like pre-test, it was somewhat like this kind of a structure, which is none other than mitochondrion. Mitochondria. So it is H. Is the option H? Yeah, there it is. So moving ahead. So here, just this question is based on amino acid. Just as an amino acid is a monomer of a protein, a dash is a monomer of DNA. So here the option will be nucleotide, as a nucleotide consists of nitrogenous base, sugar, and phosphate chain, comprising all them to to be termed as nucleotide. DNA monomer is, and the second question is the DNA monomer is called a nucleotide. Oh, so we can see that the answer of this question is already in this question. So here we can say that uh, the DNA monomer is called a nucleotide and is made of three parts: a nitrogenous base, a deoxyribose sugar, and a sulfate group. So the incorrect fourth part is this sulfate group, as the it is phosphate group present in the DNA molecule. So as we can realize from p-test, as this question is also from that, like DNA is more stable than an RNA and therefore less reactive. So the option one will be correct in this case. Okay, and we have to justify our answer for this question. So as we can remember that RNA contains two hydroxy group, making it more reactive than DNA and less and also less stable. Okay, so moving ahead, so the next part is like, is this the, it is, this question is similar to that of the previous one as well. In previous one, it was a single stranded, but here it is a double stranded. So the according to the standardness of this molecule, which type of nucleic acid it is double stranded, so it will be DNA. And according to the types of bases in the molecule, what type of nucleic acid is it? So we can see that the bases that are present is uracil, adenine, guanine, and cytosine. So as it is uracil is present, which is a part of RNA, but it is a DNA, so there must be a thymine, but it is not present here. So due to uracil and adenine and adenosine, this is RNA. So here we can take RNA. So what is paradoxical about this image? The paradoxical for this image is that due to this double stranded of the molecule, let's take the language. The molecule is DNA, but due to the presence of thymine base, okay, sorry, uracil base, it is RNA. Okay, moving ahead, according to my answer to question 11, what do delta and epsilon represent? So, based on this question, what this delta and epsilon will be. So, here we can see that uracil will be combined with will form hydrogen bonding with adenosine. So it will be A, delta will be A, and epsilon, as it is a DNA, double stranded DNA, so adenosine will form hydrogen bonding with thymine. Therefore, A and T will be the correct option, which is option A. Now, according to my answer to question 12, what bases do delta and epsilon represent? So, as we can see that here we have marked it as RNA. So here the delta will be adenosine, which will also be the case for DNA. But for RNA, the epsilon will be uracil. Therefore, the correct answer will be A and U for this case. So for moving ahead, use the following diagram along with the previous to answer the following two questions. Okay, so there are two upcoming questions which I have to answer based on this image. So here we can see it is not a deoxy sugar. It is a oxygen-rich sugar, which is due to the presence of this hydroxyl group. In deoxyribose sugar, this will not be present. This will be hydrogen, but that is not the case here. Therefore, it is a ribose sugar, so which is generally present in RNA. Therefore, this is a sugar for the previously discussed nucleic acid. There is a hydroxyl bonded to 2,3,5 carbon. What type of nucleic acid is it? So it is a RNA-based nucleic acid, as due to the presence of this hydroxyl group, making it oxy sugar, which is a which is present in RNA. So what conclusion can be drawn from my findings? Let's see. DNA can sometimes include uracil, which is totally impossible. The backbone of DNA can sometimes include this type of sugar. No, this type of sugar can never be present as a backbone of DNA. The amount of hydroxyl group does not matter. It matters because hydroxyl group defines the stability and whether it is RNA or DNA. And RNA can be sometimes be found as double stranded. So it might be the case as we can remember from the previous pretest that DNA can also be found as a single stranded. So here RNA can sometimes be found as a double stranded. So here the ordinary flow of information in a cell is based on, so this question was based on the similar question which was on reverse transcription, which was RNA to DNA, which I have marked wrong last time. But here, but after studying that central dogma of biology, so here it will be from DNA, it will convert to RNA. Further, it gets converted to protein. So this will be the correct order for this central dogma of biology. The next question, which is process that takes place at the first arrow is, and the process at the second arrow is. So, as we remember, the first this DNA to RNA is none other than transcription, which is present in this option, and further RNA to protein conversion is none other than translation. So the option 3 will be the correct in this case. So here we will see which statement about protein is correct. An alpha helix is indicative of primary structure. No, it is not correct. The beta sheet is indicative of secondary structure, but this alpha helix is what it is indicative of secondary structure, so it is also not correct. Multiple protein interacting is indicative of tertiary structure. This might be possibly the correct answer. Let's go with this question, this answer, as I totally forgotten. As the similar answer question was there in pre-test as well, but I totally forgotten what the answer was. So it is, I am going with this third option. So moving ahead, we can see that there is this combinations, and here the first question is a DNA template strand contains this sequence 5-GAG, AAC, TAT, AGT, CTG, 3 dash. What is the complementary strand of mRNA? So for complementary strand, we have to start with 5 dash will be 3 dash, G will be C, A will be U, G will be C, A will be G, A will be U, A will be U, C will be G, T will be A will be U, and T will be A will be C, A will be U, G will be C, T will be A, C will be G, T will be A, and G will be C, 5 dash. So the correct option here is option B, which is only matching with the complementary strand of mRNA. So now moving ahead, next question is your mRNA strand of choice must be reversed before converting to amino acid. It must be written as DNA again. No, complementary to the answer of to the previous question. No, as tRNA. No, it is also not correct. All these three are incorrect. But this the it will be written as from 5 dash to 3 dash. It is the correct option. So here it is, what protein sequence does this mRNA code for? Notate each protein as in three letters abbreviation and separate each protein with a hyphen. So here we can see that mRNA complementary mRNA strand is CUC. Well, let's check where it is CUC. It is LU. Let's type LU. Further, it is UUG. Let's see where is UUG. UUG is again LU. Now again AUA, where is this AUA? AUA is here, which is ILE. Further UCA, where is UCA? First letter is this, UCA, SER, then last is GAC, where first letter is G and second letter is A, this will be here, and it is ASP. So according to me, this is the correct answer. Let's check whether it is correct or not. It will be at the end of the session. So here we can see there is some more questions like a mutation, and these are two are the last ones. So a mutation causes an amino acid to change to stop codon, which is the effect of the resulting polypeptide. As we can remember that amino acid to change to a stop codon is the property of nonsense mutation, which is truncated and non-functional as well. So here the correct option is truncated and non-functional, as it is a it is the property of nonsense mutation. Now here we can see a second part, mutation causes that stop codon to change into a codon that is different from the original. What is this mutation called? So this was not none other than missense mutation. So here the correct answer will be missense mutation. So let's sum it and check how many answers are correct. So let's view score. Okay, so here we can see that previously in pre-test A, pre-test I got 14 out of 25, and here in post-test I got 19 out of 25, which is a significant improvement, I can say, for a student of non-biology background. And with this all totally different question set. As we can see here, this question is correct. Cytoskeleton is represented by G. This is cytoskeleton. Label F is none other than centrioles, and which is responsible for cell division. F is this part. Okay, okay. This is the wrong answer. Here it is the orderly represented by D is mainly found in. This D is mainly found in. It was the cytoplasm and the endoplasmic reticulum. Okay. Oh, I have given the name of that. That's a major the, mistake that I have done. It was not. It was the endoplasmic reticulum, and cytoplasm. Further, we can see the main function of Golgi apparatus represented by label. So, as we are right, this K is somewhat called as Golgi apparatus, and the main function of this is sending and receiving modified proteins. I have done it as lipid synthesis, which was the case in earlier in pre-test, which was totally different from here. Okay, the ordinary C, label C is present in. Okay, it is none other than nucleolus, which is present in the nucleus, which is present in both plant and animal cell. So here, this answer is correct for me. And the ordinary labeled. This question is totally similar with that of the previous case. So it is H for this, which is mitochondria. So moving ahead to the later section, like as we can see here, that just as an amino acid is a monomer of a protein, a dash is a monomer of DNA. It is nucleotide which it consists of deoxyribose sugar, nitrogenous base, and phosphate group. So the correct answer is nucleotide, as it is also evident from this question as well that our DNA monomer is called nucleotide. So moving ahead to the next question, DNA monomer is called a nucleotide and is made up of three parts: nitrogenous base, deoxyribose sugar, and a sulfate group. Which part is incorrect? So we know that the sulfate group is not present. It is a phosphate group. Therefore, sulfate group is the incorrect option. And this question that DNA is dash stable than RNA and therefore dash reactive. This is the similar question to that of pretest, which is DNA is more stable than RNA and therefore less reactive. Therefore, it is a correct option that I have chosen. So to justify our answer for the previous question, it is like I have written RNA contains two hydroxyl groups, making it more reactive than DNA and also less stable. But yeah, it is somewhat incorrect. Actually, RNA contains three hydroxyl groups, which is two more than DNA. Sorry, one more than DNA. Therefore, it is the correct answer is RNA has one more hydroxyl group than DNA, which makes it less stable and more reactive. So I have written it somewhat incorrect here. Moving forward, that this based on this image, that we can see that it is a double-stranded structure. So according to the strandedness of this molecule, it is the nucleic acid is DNA, so it is a DNA type nucleic acid. But due to the presence of uracil, it is a RNA type nucleic acid. So the second question is according to the type of bases in the molecule, what type of nucleic acid is due to the presence of uracil? It is RNA. So what is paradoxical about this image? I have written that due to double-strandedness, the molecule is DNA, but due to the presence of uracil base, it is a RNA, which is somewhat similar to that it is provided here, but somehow it has not given me the correct marks. So as it is written, double-strandedness is indicative of DNA, while the presence of uracil indicates RNA. Based on this two piece of information, it is inconclusive. So moving ahead, according to my answer to question 11, what does the two delta and epsilon represent? As my answer here is DNA, so the delta will be A and epsilon will be T, as thymine is present in DNA, therefore epsilon will be T and delta will be A. So answer will be A comma T, which is correct in my case. Moving ahead, like according to my answer to question 12, what basis to do delta and epsilon represents? So we can see here the delta will be A as it is connected with uracil by hydrogen bonding, and here epsilon will be U, which is uracil, which is present in RNA, which is connected with A by hydrogen bonding. So the answer will be A comma U, which is correct in my case. So based on this image of the sugar, we can see that it is not a deoxyribose sugar, it is a oxyribose sugar, which is also known as ribose sugar, which is present in RNA. So this is a component of RNA. So the question is, this is a sugar from the previously discussed nucleic acid. There is a hydroxyl bonded to a 2 dash, 3 dash, and 5 dash carbon. What type of nucleic acid is it? It is none other than RNA. Followed by what conclusion can be drawn from our findings, as we can say that DNA can never include uracil because it has thymine as a base in place of uracil. The backbone of DNA can sometimes include this type of sugar, which is totally not the case, which is present in RNA, and it is not a deoxyribose sugar, therefore it is not present in DNA at all. And the third option is the amount of hydroxyl groups does not matter, but it really matters in terms of stability and reactivity. Therefore, it is not. It is also the incorrect option. And the last option, which is RNA, can sometimes be found as double stranded. It is the correct option. So the followed next question is the ordinary flow of information in a cell is, as we remember from the part A question, that it was that asked about reverse transcription, which was conversion of RNA to DNA. But here it is a forward order. So first will be DNA to RNA, then RNA to protein, which is a central dogma of biology, followed by the question, the process that takes place at the first airways and the process at second airways. As we know that DNA conversion of DNA to RNA is transcription and conversion of RNA to protein is translation. So option C is correct and I have correctly answered this. Followed by which statement for protein is correct. So the and alpha helix is indicative of primary structure, which is not the case. It is sequence of amino acid, long chain of amino acid. It is a primary structure. Beta sheet is indicative of secondary structure. It is correct option, which I have totally forgotten and marked the incorrect answer. So moving ahead to the next part, as we can see here, the sequence and the respective amino acid is given here. So based on this, the question which is our DNA template strand contains this sequence which is 5 dash GAG, AAC, TAT, AGT, CTG 3 dash. What is the complementary strand for mRNA? So we have to go that 5 dash will be the 3 dash. 3 dash is same in all the cases. G will be connected with C, A will be connected with U, and G will be connected with C. So based on this, only this three connections, we can conclude that option C is correct, as in all other cases, the answer is GUG, which is not correct in this case. Followed by your mRNA strand of choice must be reversed before converting into amino acid, that is, it must be written as DNA again, so it is incorrect option. Complementarily to the answer to the previous question, it is also not correct. As tRNA, it is also not correct. But the option 4 from 5 dash to 3 dash, it is correct in this case. Here it is written, the next question is what protein sequence does this mRNA code for? Notate each protein as its three-letter abbreviation and separate each protein from with a hyphen. So I have somewhat given the wrong answer, and it is true that I have given the wrong answer as we have to move from 5 dash to 3 dash, and the complementary will be GAC. We have to check for GAC, where GAC is the first letter is G and A is here, so GAC will be written as ASP. No, no, sorry, GAC, not GAC, it is CAG. Like we are moving from this to this, so it will be CAG, and CAG will be C here, this A here. CAG will be written as GLN, therefore here it is written as GLN, followed by ACU. ACU is written over here, which is THR, and this is the case here, THR, followed by AUA. AUA is none other than ILE, and then further it is GUU, and then GUU is over here, which is VL. Here it is written VL, and the last is CUC, which is none other than CUC leucine. So the correct answer is this. However, I have learned the concept wrong, so the answer is incorrect. So followed by this is the last part. A point mutation causes an amino acid to change to stop codon. What is the effect of resulting polypeptide? As we remember that it is the property of nonsense mutation, which is truncated and non-functional. Therefore, the option B is correct, and I have given it correctly, answer it correctly. And the last question is, second point mutation causes that stop codon to change into a codon that is different from the original. What is this mutation called? It is missense mutation. Actually, the mutation which is similar to the original is silent mutation. but here it is different from the original, therefore it is a missense mutation. So by this question, I would like to end the video. Thank you and have a good day.